traveled to East Lansing, Michigan, to Michigan State University's campus, and observed the black-capped chickadee birds outside of C7 Holmes Hall. We chose the black-capped chickadee birds simply because it was getting a bit colder out, and if you know anything about birds, they don't like dealing with the cold. They like flying south for the winter. However, chickadees are a non-migratory species, so we chose this group of birds specifically because they wouldn't go south, so we could actually get reliable data during the cold winter period.
When we began to brainstorm on how we could document communication behavior between birds and humans, we came across a study completed by Timothy Boswell and his colleagues, in which he studied the relationship between the neuropeptide Y gene and hunger through experiments with broiler chickens. He had two groups of these chickens, those who were able to feed freely and those who were on a restricted feeding schedule. And after he measured the levels of NPY mRNA produced in the brain and concluded that higher levels of the NPY protein are produced in feed-restricted chickens, indicating that there is a link between the NPY gene expression and hunger.
My team and I then asked, what behavior can we observe that is related to hunger? And can we find evidence of that behavior that would indicate the presence of the NPY gene?
We determined that we can observe aggressive behaviors in correlation with the NPY gene and hunger.
And we'll be doing this by observing aggressive behaviors in birds outside at the bird feeders. And we will also be doing this in humans by observing their aggressive behaviors in the various dining halls.
Let's get started!
We left only one bird feeder hanging, and we took the rest off to enhance competition during feeding times. We hoped this would stimulate aggression in the chickadees, and we defined the aggressive behaviors we were observing as flapping wings at another bird, chasing it off, or guarding of the feeder by sitting on top of the perch. All of these behaviors were based off preliminary data.
We then traveled to dining halls on campus to observe human aggression during feeding time and looked for defined behaviors such as crossing arms, tapping foot or jittering leg, and facial expressions such as a wrinkled brow or pursed lips.
Here we are observing the guarding behavior of the bird on top of the perch.
Here we have a chickadee on the feeder, and he's chasing the other bird off.
Here we see a chickadee chase another chickadee off the feeder while flapping its wings. In the dining hall, we observe subjects jittering their leg or tapping their foot while waiting in line.
Here we have a subject crossing their arms while waiting in line.
Here, we see that this fellow is expressing some aggressive facial expressions. Here, we have our predicted PCR figure. As you can see, this is the KB plus ladder. This is a molecular weight ladder, so it is already automatically determined. Here and here, we observed fluorescence at 215 and 317 base pairs.
Here is the compiled data over the course of our research of the aggressive behaviors observed at the disturbed feeder. Behaviors include chasing, guarding, and flapping wings. Here is where we ran our ANOVA unpaired t-test to produce our significance and error bars.
The three graphs here are displaying the express or null behavior for each three of the observational behaviors we were looking for in humans. We ran the ANOVA unpaired t-test to determine significance and error bars for all three sets of data.
Due to limitations in time, we were unable to run PCR tests in order to identify if the NPY gene was present in our subjects. However, through statistical analysis, we were able to calculate our significance for the observations made at the disturbed feeder and of the humans made at the dining halls. Significance values at the feeder were calculated at 0.05 and of human data 0.01.